Welcome back to What Did I Do in Lab Last Week, where I tell you guys what I did in my biochemistry lab last week. So this one was the second part of the DNA transformation. So last week my lab partner and I made a bunch of different DNA bits where we took viral DNA and put it into bacterial plasmids. And this week we were transforming those plasmids into viable E. coli bacteria. So what we did, we got all the little tubes of the DNA we made and got a thing of live E. coli, and then we put the different DNA plasmids into the E. coli, left them on ice for a little bit to get them to chill out, and then we did what is called heat shock transformation. So after the plasmids are made, we need a way to actually get them into the bacterial cells, and the way we did that was by heat shock, and we don't really know how this works. Scientists don't really know, but we do just know that it does work. So what happens is you put the DNA into the tube with the bacteria, and then you take that tube, put it in hot water for 20 seconds, and then take it out, and somehow the DNA gets into the bacteria. And then after the heat shock, the bacteria are a little bit freaking out because what did you just do to me? So then we have to put them in an incubator for an hour to get them back to normal and stop freaking out from the heat shock. Then after we let the bacteria cook for an hour in the incubator, we have to plate them on a bunch of different plates in order for them to grow. So that was on Friday, and since I'm on a reading break right now, I can't go check on my bacteria until next week to see if any of them actually grew. Last year, me and my friend did this basically exact lab in genetics last year, but a bit more complicated. It was like the entire cloning process. This was just the DNA ligation and transformation. But unfortunately last year, neither of our bacteria grew, so hopefully they actually grow this year. Yeah, I will put an update on what my bacteria plates look like when I actually go see them and get the results of this lab.